Hi, thanks for tuning in to a new episode of the Doctor Podcast Show, and I'm your host, Dr. Robert Sayour. Today, we're discussing a very important topic. It's a bit of a sad topic, but it's also extremely important. We're going to discuss brain tumors and brain cancer in children in the pediatric age group. And we have with us today a great guest, one of the world's leading experts on this subject, who's done some incredibly innovative research to treat this horrible condition. Our guest today is Dr. Elias Sayour. Dr. Sayour is an associate professor of neurosurgery and pediatrics at the University of Florida in Gainesville, Florida. He's also the assistant dean for clinical research at the University of Florida. He's the director of the Pediatric Cancer Immunotherapy Initiative, and he's also the principal investigator of the RNA engineering laboratory, which we'll get to in great detail. By the way, if you're enjoying this episode, please click on the like and repost button. And of course, please subscribe and follow Dr. Podcasts. We always appreciate that as well. So, Elias, thanks very much for coming today. I know you're extremely busy. You're working very hard, so I appreciate your taking the time to join us today to explain what you've been doing recently and this incredible article that you recently published about treatment of brain cancer in children, which is very new and innovative. Well, thank you very much.
Thank you for having me.
Yeah. So tell us about your education after the usual 4 years of medical school. What additional training did you get, and for how many years was that?
Still many years after medical school. 3 years of pediatric residency in New York City. Then I did 3 years of pediatric oncology fellowship at Duke University, and was crazy enough to stay on there as part of graduate school to finish a Ph.D. And then as I was finishing my Ph.D., but really towards the end, came down here to the University of Florida to start on as faculty.
Wow, so many extra years of training. What did you get your PhD in?
Experimental pathology.
Wow. So that's how you got interested in this. So you're originally from New York City. What got you down to Florida other than the great weather?
It's a great question. I think I ask myself that frequently too. How does a native New Yorker end up in Florida now for over 10 years? Um, well, when I was at Duke, I met a tremendous individual, Dr. Dwayne Mitchell, who I met him at a time in my life where I was really having a lot of challenges with the field of pediatric oncology, a lot of challenges with the outcomes. To this day, I really struggle with the fact that we have to treat 10 patients with poisons only to cure 4 or 5 or 6, depending on the disease. And so I felt myself then and now often breaking the first rule of medicine, the do no harm rule. And so this was a real struggle for me, and wanted more meaningful therapies. And this field's pediatric oncology, certainly for solid tumors, for brain tumors in particular, there have been no new therapies for many decades.
Right.
And as I was having these feelings, I met Dr. Mitchell, and he spoke a different language, a language of research, language of the immune system, and using the body's own natural immune system to fight cancer. And I was bitten by the research bug pretty quickly. This whole new world that I kind of knew about, but didn't really understand how far along his group and other groups were in the field of immunotherapy. And this was really before the big immunotherapy boom of the last 10, 12 plus years. And so as I got to know him and be more familiar with that work, I wanted to learn as much as I could. And so I went back to graduate school, and in some crazy way, graduate school and research saved my clinical career, because it really allowed me to continue to practice in a way that at least satiated this need to feel like I was doing something more. And I still struggle today, not knowing if this work is truly going to matter, but it has at least provided some nourishment to this need to feel like we're trying to do something about these bad outcomes. And even the good outcomes are when you have side effects, children that can't walk, talk, hear, treatments for brain cancer, even the cures are hard to feel that good about. So this has been important work, and we're hopeful that the immune system can be a new modality to elicit not just improved responses, but long-term protection. That's the beauty of the immune system. It protects people lifelong. Chemotherapy's on and then it's off, and then you're dealing with side effects.
Right.
The whole point of immunotherapy is, can it provide long-term protection? And I believe it can.
And minimize the depressing side effects from chemotherapy, radiation, surgery.
Absolutely. Absolutely.
Right. So you're performing incredible work. We really appreciate it. Now, tell us why young children get brain cancer. Usually when we think of cancer, we think of older individuals, senior citizens getting that. In the last few years, there's been increased number of people getting colon cancer at a relatively young age. But why do young kids get this? Don't they have very strong immune systems? And isn't the immune system supposed to find and kill cancer cells before they can grow?
Great question. And I want to answer that question. I also realized I didn't answer your previous question. I met Dr. Mitchell and I was recruited here to the University of Florida because he moved from Duke to Florida. And it's
Oh,
been
okay.
just an incredible opportunity to work here and partner with him all of these years. Um, Children getting cancer is not natural. I mean, in fact, nobody should get cancer.
Right.
But older people tend to get cancer in some ways due to aging cells, mistakes, mutations that happen, sometimes due to environmental triggers, the complexity of how genetics, aging, and environment interact. But children, these are not aging cells. These are young cells. And when a child develops cancer, it is very different from an adult cancer. And this is certainly true for glioblastoma, which is the most common malignant brain tumor. Children can develop this tumor. So do adults, but they're very different. Even though we call it the same thing, glioblastoma, these tumors molecularly are very different. And so why are they different? Childhood tumors are what we refer to as more epigenetically driven, meaning it's developmental genes that are supposed to turn off during childhood that somehow turn back on again. That dysregulation that, when a fetus is growing, the developmental genes are on and they're, growing really quickly, that's supposed to turn off. And when it turns back on in a dysregulated manner, that can lead to oncogenesis cancer. Adult tumors tend to be more genetically driven. There tends to be more genetic aberrations. And in some ways, this is an oversimplification to say that adult tumors are genetically driven and pediatric tumors are epigenetically driven. It's very, very complex, and it is a complex interplay of environmental cues and genetics as well. But as an oversimplification, these tumors, these pediatric tumors, are certainly much more about how genes are regulated than the genes in and of themselves. And part of the complexity of all this is not even the tumor cells, but the tumor ecosystem. The tumor ecosystem is different across childhood cancer and adult cancer. And that ecosystem is not just the tumor cells themselves, but the shields, the stroma, that the tumor is growing in. The immune cells also form part of this ecosystem. And one of the things our lab is trying to do is really understand the different factors of the cancer ecosystem between childhood cancer, adult cancer, individual cancers. Every individual has its own unique immune system and also unique ecosystem within cancer. And in many ways, that personalization and understanding cancer at that personalization, not just at the tumor cell level, but the tumor ecosystem and the immune system as part of all of that, is really important to redirect it, reprogram those environments, and ultimately unlock the effects of the immune system against each individual cancer.
Right. So basically, these cancers occur because normally brain tissue is growing very rapidly in young children and developing, but somehow some cells start growing out of control and multiplying too quickly, and they have abnormal proteins. Now, normally the immune system is supposed to find abnormal proteins and
Yeah.
attack them and get rid of the cells, but something goes wrong here. There's a misread by the immune system, and it lets these abnormal cells grow and proliferate?
Yeah, another great question. I mean, look, I believe fundamentally that the immune system is killing cancer all the time, killing cancer all the time in children, killing cancer all the time in adults. And I have always struggled with this idea that cancer is a self disease. I mean, we hear that all the time. It's, your our cells are being, you know, um, are growing abnormally and turning into cancer. The reason I struggle with that is the immune system is smart enough to find our normal cells that are infected with virus. So a virus infects a cell, it hides in our cell, it hijacks that cell, and that cell is still 99.9% self. And yet the
Right.
immune system knows to kill it, knows that that cell is infected with virus, and knows to kill it. I would argue that the cancer cell is far more foreign appearing than that normally infected cell with virus. And yet the immune system, when a cancer has developed, is often not involved at all, or if it is, does see this foreign thing as self. And so why is that? It's complex. And I can address some of that complexity by saying that it's a lot of evolution. These cancers, as they're growing, are evolving in a way that tricks the immune system. And what I mean by that is the immune system is not just a killer force. It is. It is a potent killer force, but it's also a healing force. Anytime
Right.
we cut ourselves, anytime we develop any wound, the immune system is involved in wound healing. That's why we struggle with chemotherapy, right? We get rid of immune cells, and now there's poor wound healing, and patients can develop sores, and they're at risk for infection. So that wound healing is a critical feature of the immune system. And the best way that I could put it is in the context of glioblastoma and related tumors. The immune system doesn't see cancer as this foreign thing as much as it sees it as it's a wound it has to heal. And as the immune system is trying to heal that wound, it creates an environment that is actually much more pro-tumoral. And the tumor now can invade, this chronic inflammatory phenotype that allows the tumor actually to invade even further. And the immune system comes in and tries to heal it, allows the tumor to invade further. So it's a vicious cycle. And our hope is to first break this cycle and reset this response in a way that can finally now educate the immune system that this is foreign. The problem is, even when you do that, the cancer can be so far gone. I mean, it truly is a race.
Right.
And in some cancers, the immune response is keeping up in the race. Certainly this is true in melanoma, and you can maybe give it a drug like a checkpoint and give it a little bit of a boost. But in many of these cancers, the immune system isn't even off the starting line. And if anything, it's going in reverse. So those are the real challenges we're facing in many of our cancers, like glioblastoma, and really provided the background for the work that we're doing, trying to develop new therapies to try to break these cycles, reset these environments, and induce responses that are more in favor of the immune response.
Right. You know, what you're saying reminds me of a documentary I watched this past weekend about octopuses. Octopuses can change their coloration within seconds to match their environment. And they showed a segment where a shark came by and an octopus changed its color to look like the rocks just behind it. It's almost like these cancer or tumor cells camouflage themselves so that the
Oh,
immune
yeah.
system just kind of goes by and says, "Oh yeah, you look okay. We'll let you go and move on." So it seems like there's a camouflage going on, and the immune system just kind of ignores the cancer cells. Is that a fair analogy?
Oh, absolutely. That is a very fair analogy. Um, and they have even more mechanisms to camouflage. I mean, there's so many different ways they leverage to mask themselves and to leverage the local regional milieu, their environment, to drive their own continued growth. And as they do this, they reform this environment so that the cancer is actually naturally selected to be the only thing that ends up surviving. And yet it's something I never understood. It's really for one end, and the end is death of the cancer, death of the host, which is very unnatural. I mean, that is unnatural. That's just not the
Right.
way. So I mean, I don't use the word evil lightly, but to me, cancer is evil. I mean, it is the only end it has is death, and that's yeah,
Right. That goes against nature and evolution because it's supposed to keep its host alive so it can keep growing,
exactly,
yet it
exactly.
kills the host. So it doesn't make that much sense, but hopefully you'll figure it out. Now, traditionally, as you mentioned, these glioblastoma cancer tumors in children are treated with surgery, radiation, chemotherapy. How successful has that been over the years and actually decades that it's been used?
So I mean, these are the mainstays of therapy. Surgery and radiation have really been the mainstay. Certainly for pediatric high grade brain cancers, glioblastoma, those are the only things that have worked. Surgery, radiation. In pediatrics, chemotherapy really hasn't worked.
Hasn't
Uh,
worked.
Chemotherapy one drug has worked in adult glioblastoma, temozolomide, but admittedly not that well. And I would say outside of local control with surgery and radiation for pediatric tumors, that there's just not much else. And that buys you time. And for adult patients, using all three of those things, the median overall survivorship is still about 15 months.
Mm.
And for children, it's not much longer, to be perfectly honest.
So
So
it's almost 100% mortality, basically.
it's, yes. Now, the field has shifted to really molecular diagnostics, meaning if you have certain mutations, molecular features, we know that your outcome is either going to be better or worse. I will tell you for some pediatric tumors, for some malignant gliomas, if they have a certain mutation in a certain location, the cure rate is effectively zero.
Mm.
And that, to me, that is the worst of the worst of the worst when I see these patients and I can't even provide any hope because the cure rate is effectively zero.
Right.
So the need to me is dire. Here in the United States, cancer is now the number one cause of disease death in children, and brain cancer really makes up the bulk of that. So we need new therapies. I think discovering new therapies for childhood cancer, I think will allow us to have new modalities for all cancer. And this is true because combination chemotherapy really started through the study of childhood leukemia and being able to cure childhood leukemia and leveraging that knowledge for other cancers. So, orphan disease work has been a premise for advancing knowledge and advancing therapy for other indications. And I believe this. I think if we can cure childhood brain cancer, I think we'll have a pathway to cure all cancer.
Right. So tell us now about your research and as background, tell us what nanoparticles are and lipid nanoparticle aggregates that you're using in your research and now in your treatment. Can you explain what that is and how you're modifying the immune system, modifying the tumors?
Sure. So, I mean, generally speaking, RNA, which is an incredible tool, and it's been around for decades now. It didn't just start with the COVID-19 pandemic. I mean, we were FDA IND approved before the pandemic to do this work. We've been working on RNA vaccines for a long, long time, as others have. And
Right.
one of the reasons it got approved as quickly as it did is because of all of this research over many years, decades. Now, the one problem with RNA is it's not very stable. RNA degrades very easily. So it's a very powerful tool. RNA is like software, it's information. And the ability to program software and information can be used to basically program the human body. Now, the problem is delivering that information is really the key because the information needs a delivery vehicle. RNA will degrade very quickly without that vehicle. And so what has been used for the COVID-19 vaccines is something called lipid nanoparticles, basically fat droplets. It's an oversimplification, but basically fat droplets to protect the RNA from degradation. So the RNA is inside these lipid spheres, and that's injected intramuscularly to elicit an immune response. Now, that works very well for COVID, but here's the problem in cancer. Cancer does not have, glioblastoma certainly doesn't have something like spike protein that is so profoundly foreign that that is going to elicit a really strong response.
Right. The
And even
spike
if
protein is present on the COVID virus.
it's present on the COVID virus, and even in the preventative setting, the expression of that protein is very foreign. Like that elicits a really strong immune response. Now, even though I say cancer is foreign and it has foreign signals, it's still, these signals are not as strong as virus. And the truth is, trying to get immunity against something that's already happened means you have to be much quicker. Okay, even the COVID vaccine, you have time to boost it because you're preventing something. But cancer's already happened. You don't have time. You don't have weeks. You don't have months. You need a response immediately. And that response really has to start to stymie and overcome the evolution of that cancer. So we designed something that is a little different, unorthodox for this field, and certainly something we feel can harness the immune response much more rapidly. And so instead of creating these lipid spheres to protect RNA, and there's only so much RNA you could package into a sphere because RNA is negatively charged. So
By the way, sorry to interrupt, how small are these lipid or fat globules? These are tiny microscopic things. They're not a big giant thing, right?
they're bigger than the typical particles used. So these are about 200 to 400 or 500 nanometers in size. So still tiny at that level, but at the nanoparticulate level, actually quite big.
Right. But it's much smaller than something you can see. Just for
Oh
the
my gosh,
audience, this is
incredibly.
like
I mean,
microscopic.
oh yes, I mean a cell is much larger than that. I mean, double, triple, quintuple in size.
So these
So the
are about a
still,
quarter the size of a human cell.
I think that's fair. Yeah, about. So these particles that we're making, yes, they're bigger. And one of the reasons they're bigger is we're actually creating these onion-like structures. So the RNA basically tethers to a particle and another particle surrounds it, and then there's another layer of RNA. And so kind of these onion-like clusters form. And some of these clusters start aggregating around other clusters. And so you now have, instead of kind of these single particles like I described for COVID, you kind of have these aggregates. And it has been shown for viruses. Viruses actually do this. They cluster, and that actually is more infectious than, single viral particles. So we kind of use that kind of as a means to make more strongly infectious or antigenic cancer vaccines by creating this onion-like clustering effect, and now not injecting it locally in the muscle, but injecting it intravenously. Basically tricking the human body into thinking there is a very, very serious infection that is happening immediately that the entire immune system needs to respond to. And sure enough, we see that. Within a couple of hours of injecting this, just about every immune cell that we can find is being trafficked or mobilizing to this significant danger. And the danger is really your own cancer. That cancer tries to mask itself. We're trying to unmask it, make it look like the predator, the threat that it is.
It's getting rid of the camouflage, basically.
Essentially, yeah. That's why it's a good analogy. The octopus analogy is a good one.
Yeah. Now, the thing is, you mentioned RNA or messenger RNA is basically software. It programs certain proteins. How did you discover which mRNA or RNA to use? How do you know what the RNA code should be?
That's a great question. And that is the challenge in cancer. because I described how different every tumor is from another patient's tumor within the same tumor, which is crazy. Within the same tumor, cells at the surface are different from cells at the core. Within the same tumor, there's competition. There's different looking tumor cells.
Hmm.
Because of how different tumor is within each patient, we decided to take all the RNA, basically everything that's expressed in that tumor, we are vaccinating against. And we're allowing the immune system to parse through all the information in the patient's tumor and target what we refer to as the heterogeneity of the disease, basically how different this is across the entire tumor can be picked up by the immune system. We can use these vaccines to educate the immune system to develop not just a single response against a single target, but what we refer to as a polyclonal response, a response against many, many targets simultaneously, which I think you need. We're not going to be able to cure cancer unless we're targeting many, many, many targets at once.
Right.
Now,
So the question is, do you take the RNA from the tumors and then create a protein or an antigen that then allows you to create new RNA, or are you reverse engineering the protein antigens from the tumor to make the messenger RNA or
yeah,
RNA
good
that you
question.
need?
So from as little as 100 biopsied cells, we can extract all the information and amplify all that information. We can literally make a cDNA library and amplify mRNA whenever we want as a renewable source of vaccination for that patient.
How long does that take to create that RNA?
Oh, I mean, we can do this in a week or two.
Really? Wow.
Yeah, it's a little longer for patients because of all the quality controls, but in the lab, we could do it in a couple of days. So about a week or two in patients when you consider the quality control. But
And another question, sorry to interrupt, is the RNA different from one patient's tumor to another? Or are
yeah,
there some that are the same and then there's a few outliers that are different in patient to patient?
it's really quite different. I mean, I mean, there's some things that are similar, but when you really look at the individual level of what's driving these tumors, what are the ideal targets, perhaps, if you were to look really more deeply, they're quite different. And we are thinking of potentially refining this to really identify the most foreign looking information, across a patient's tumor, profile that and just synthesize those RNAs. That's going to probably be a second generation approach. Right now we're vaccinating against everything, which may be enough, quite honestly. But it does seem to be quite different across these tumors. I think I understand maybe what you're getting at, which is, could you potentially make a universal vaccine that captures maybe the commonalities across cancers, and people have looked at that. And I will tell you that is a hope of mine, that as we look across every individual's cancer, not just brain cancer, but all cancer, we will identify what we refer to as hotspot mutations, hotspot regions that are maybe common across certain cancers or certain subsets. And now you can imagine a scenario where we can build a warehouse where we make an RNA for every hotspot that we can find across all cancer. I mean, there would be hundreds, if not thousands of these, but we can have them all pre-made. And then if a patient comes in and has, XYZ, ABC hotspot, you can just pluck them off the shelf from your warehouse, make your RNA vaccine, vaccinate that patient. That's an off-the-shelf vaccine, but quite personalized to that patient. And so that is my hope eventually, to make
Now,
this really commercializable.
right. Now, the RNA that you inject into the vein intravenously, does that then produce a protein which the
Yes.
immune
Yes.
system senses? Is that
Yes.
how it works? So
Exactly.
the immune system doesn't really attack the RNA, it attacks the proteins that the
Exactly.
RNA codes for?
Exactly. The RNA so this basically transfects cells all over the body, and that information is turned into protein that is presented to the immune system, and the immune system basically seizes on those protein signals to basically fight the cancer.
Right. Now, what you're doing is this tumor was kind of isolated and hiding from the immune system, but now that you've put it into the entire body, the immune system can't ignore it anymore. It
Exactly.
sees these proteins. Is there a risk, though, that the immune system might overreact to these proteins and
Yes.
create all sorts of cytokine storms and inflammatory cycles that might be damaging to other organs in the body?
Absolutely. I mean, this is something that is very important for people to be aware of. As much faith and belief I have in the immune response, and it is, I do believe it is our guardian angel to prevent cancer from happening, and I do think it can be used to and there is evidence for this, be used to protect people lifelong from cancers they've developed. Uh, the immune system is like fire. Okay, fire is a great thing. You can control it. You can we can do also humanity's done all sorts of amazing things because we've been able to control fire. You don't control fire and horrible things can happen. And so this is the immune response. If we push it too strongly, too fast, it can self-amplify in a way that can be quite harmful and deadly. And this has happened in some immunotherapy trials. And this is a terrible fear of mine, honestly, as we trailblaze in developing a new technology that's never been done before. And we are seeing immune effects, and we are seeing cytokine response syndrome, and we are seeing kind of at low doses, which is creating this fear of, well, how high can we go? How many doses can we give before it's to the point of no return? How safely can we boost this? I mean, we're grappling with this right now. I don't know the answers to any of those questions. And another one is, how do you combine it? Because you probably want to combine it with other therapies to maximize immune response. So this is really a new frontier. I think it's exciting, but believe me, there's a lot of trepidation that I have about it. There's a lot of trepidation a lot of people in the community have about it because we don't want to hurt anybody. And
Right.
yet we know that this is a type of thing, the immune response, that if you push it too far, too fast, can it can be fatal. At the same time, you want to see a little of these things because you want to, fire is heat. You want to see some of that, right? You want to be able
Right.
to see it and control it. Because if you don't see it at all, you worry, are you even having an effect? So it's this Goldilocks effect, and it you don't want it to be too strong, but at the same time it has to be there. You have to see signs of it, and it is such early days for the entire field. I don't know that we know in the field how do we use these tools, whether it's T cells, mRNA vaccines, checkpoint inhibitors, oncolytic viral therapies. How do you use these? How do you dose these? How do you mix and match these things in a way that can be used to provide synergistic immunotherapy? I can tell you this approach is profoundly immunogenic. It does elicit very strong cytokine, chemokine response, mobilization of cells into the tumor. We know this in the periphery, and it's early days for us, and we're excited about the immune effects, but at the same time, we're quite concerned about them as we decide how much of this can we safely give and how many boosters, et cetera. Um, in terms of autoimmunity, which is related but separate. When I think of the immune system, it's that's the early effects of the immune system, which you can self-amplify very quickly and be very dangerous. The cytokine storm you mentioned, but also as you're fighting the cancer, the immune response may spread to normal targets, and that could cause autoimmunity. And certainly, if it's happening in the brain, things like multiple sclerosis or demyelination or encephalitis, encephalomyelitis, those things could all be possibilities. But the truth is, the reason most immunotherapies have failed, certainly in brain cancer, and I would say across cancer, is not because of autoimmunity. They failed because they're not strong enough. And so I hope we didn't go overboard with our design and our intravenous route of administration. But I will say that most therapies have failed for not really being robust enough. And I hope and pray that this is safe and effective. Um, but I
Well,
do think we need immunotherapies that are more robust.
you're starting with somebody who has almost no chance to survive. And this is the beginning. We're learning how to use this new tool. Eventually, hopefully, we'll be able to quantitate it better and make it very specific for each patient. But you have to start somewhere. Now, you've done research on dogs initially, right? To
Correct. Yeah.
test this out. Can you tell us a bit about that?
It's one of the reasons I've just found the University of Florida the most incredible place for me to work, to raise a family. But the idea that I can actually take discovery and move it into clinical trials without industry, without commercial sponsorship, to me is very exciting for orphan diseases like pediatric cancer.
Wow, that's
Um,
amazing. So there's no pharmaceutical companies or drug companies involved in this?
I mean, there are companies interested in it and have licenses and options, but for the cancers I'm interested in, you really have to move it forward yourself. And in many ways, that actually creates more interest, more commercial interest. And I lament all the time, how many times have we cured cancer and we just don't know it because it's sitting on a shelf and nobody's moved it forward, or it's sitting as part of a company's IP, but never really
Right.
cared about moving it forward. And so I really do think, I mean, I believe this. Academicians, academia should play a larger role in moving forward therapies into early phase trials. And I think most places, understandably, because it's hard, most places are just waiting for Pfizer, Genentech, Eli Lilly to come over and say, okay, we're going to help you out. And I get it, it's hard. But I think when you're a place like the University of Florida and you have all the resources, then I do think it's incumbent upon us to do just that, to move these things forward. And UF has done that for gene therapies. AAV gene therapy was largely discovered and developed here. We're trying to move forward mRNA vaccines and other cancer immunotherapies, CAR T cells, dendritic cell vaccines, using the institutional capacity to manufacture pharma. So we can manufacture through our CGMP manufacturing facilities, pharma grade compounds. We have cores, though, to move forward discovery into trials through a FDA IND application. And so the FDA, they want to see large animal studies. And most companies often have to do primate work, which has its own ethical challenges of doing primate toxicology studies. So working with the veterinary school to treat dogs and cats, these are pets with natural forms of cancer. We're not injecting any cancer into these animals. They're enrolled through their owner's consent, much like a patient or a child would come in and we get a consent from a parent. These pets, we get consent, we get their tumor right there, we make the vaccine in-house here, we vaccinate the animal. And the fact that we've seen responses in dogs with brain tumors, dogs with bone tumors, we haven't published on that, cats with head and neck cancer, really it does a couple of things. It makes us feel good that we're able to help these pets, but also it really helps us move forward these approaches through the FDA, because that satisfies, at least it has for us, it satisfies kind of the small animal studies and the large animal studies that they're looking for to demonstrate feasibility, safety, toxicology. And honestly, because these animals have the cancers, and by the way, these cancers look a lot like the pediatric cancers in the brain
Yeah.
and the bone tumors, it really allows us to feel like this has a chance of helping people. We're also able to monitor these cats and dogs in a way that has served as kind of a roadmap of what we need to do in humans. And so when you're going live like this, like live, like you've got to do dogs here, get the tumor, make a vaccine, get the vaccine back. When you're going live like that, you learn so much. And we've learned so
Yeah.
much that has really helped us to really be prepared for the human trials and be primed to do that work. And so I just can't say enough good things about our partnership with the College of Veterinary Medicine and the work we've done with cats and dogs with terminal cancer.
That's amazing. University of Florida deserves a lot of credit to get all that done and
Yeah, no,
accomplished.
it's been a tremendous environment and very grateful to be here.
Now, tell us, have you treated human patients with these cancer vaccines, mRNA or RNA cancer vaccines?
So we've treated a small handful, only 4 patients to date, with these vaccines, and we found really much of the same in humans as we had seen in mouse and dog. Basically, these work very quickly to activate just about every level of the immune response, the early part of the immune response, the late part of the immune response. We see this in just a few hours from a single administration.
Wow.
I think what surprised us about the humans relative to the mouse and the dog was how the booster effects in humans were even more significant, I think, than what we appreciated in our animal studies. Meaning the next vaccine, the immune effects were that much stronger in the humans, which is exciting, but at the same time has led to these questions about, well, how many can we give? What's this Goldilocks? In terms of dose, frequency, timing. So we're grappling with all of that right now. But the booster effects have been significant, and in one patient, we saw what we thought was a massive recurrence in the tumor, and so much so that that patient went back for a re-resection, and the tissue there were no tumor cells. It was essentially all reactive immune, which I think is demonstrative of the fact that this can really elicit some pretty strong responses.
So basically the tissue was dead cancer cells and immune cells like
basically,
white blood cells and
basically,
lymphocytes and so on?
basically, more or less. Reactive gliosis, immunologic signs, lymphocytes, as you mentioned, but no tumor cells.
No tumor cells.
Yeah.
That's pretty amazing. How's the patients that you've treated done clinically?
So it's really hard to say from a cohort of 4 patients, and which have been treated with very few vaccines and I can't stress that, very low doses of vaccines. One of the beauties of this approach is, unlike other approaches, we can make as many vaccines. We could probably make vaccines lifelong for an individual if we needed to. So this is an approach that can be really very, very feasibly grown in large scale numbers for individuals, patients, repeated doses. The challenge now is trying to figure out exactly, as I mentioned, how many of these things can you give? What's the dose? But to answer your question, 2 of the patients only got 2 doses, and the other 2 patients got 4 doses. So very few doses we've given so far. The progression-free survivorship of this disease, because we took really only the worst glioblastoma, so they had to have a molecular phenotype that basically allowed us to forgo chemotherapy, because chemo doesn't work that well when you have this particular molecular phenotype. In these patients, their progression-free survivorship is about 6 months. And so in these patients, they all lived longer, about 8, 9, and 10 months, if I'm remembering the data correctly. So certainly longer than what would be expected, but also no long-term survivors meaning they all ended up progressing. And that's kind of what we're struggling with. Should we have given more? Should we have boosted them more? Really, in this cohort, we really were doing more phase 0, I would say. So phase 0, trying to identify kind of a dose that we felt would be biologically active. And we're surprised to find that at every dose that we had administered, there was biologic activity. So I think there's some promising signs here that this could work, but I would just really highlight that there is a lot more for us to do, a lot more for us to learn. But I think if we do learn that, what's the maximal tolerated dose, and how can we give more and more and more and more to maintain responses? How can we combine this? I think the future is bright with the knowledge base that we have now.
It sounds like you've figured out how to get the immune system to recognize the cancer and attack it, but there's a lot of fine tuning that still needs to be done.
Yes, I think that's well stated. Lots of fine tuning. And and we want to be careful, right? We certainly want to be very delicate about this because we don't want to push forward too fast and too quick with so many higher doses and frequent doses that we jeopardize the entire platform.
Right. In the patients that you treated, were their tumors debulked first with surgery or radiation, or you just
They were. They were. But even with that, I mean, glioblastoma is such an awful disease. I mean, people used to do hemispherectomies, like take out half the brain and the tumor would recur on the other side. So the truth about this tumor is even with complete surgeries and radiation, the tumor is microscopically invaded the entire brain. I mean, that is the unfortunate reality of this tumor. And so you need some type of systemic therapy for that.
right.
Yeah, and we just don't have it. I mean, to me, I'm an oncologist, and I feel like I fail again and again and again because it's my job, it's our job to have systemic therapies, right? Surgeon, they do the local control. Radiation oncologist, they come in with further local control. We're supposed to provide the systemic therapy that flushes that disease out. And
Right.
we just don't have the tools outside of chemotherapy, which doesn't even work for my patients with pediatric high-grade glioma. So, it is really surprising for me in 2024 with all the discoveries that are being made, AI and next generation sequencing and new imaging modalities and understanding biology and cancer at such unprecedented levels. It is honestly shocking to me that our mainstay of systemic therapy for this cancer and many, many others is still kind of poison, chemotherapy.
Right.
So, I think we need new therapies. I think there's a lot of hope, not just in this approach, but other immunotherapies that are being developed.
Right. I
As
know
really new
melanoma
treatment.
has immunotherapy that works pretty well. I actually have a few patients who've
Yes.
survived many years. How is what you're doing different than the melanoma immunotherapy?
Oh, very different. I mean, for example, Moderna has a phase 3 trial for an mRNA vaccine in melanoma being combined with those drugs, those checkpoint inhibitors, which work quite well in melanoma. And
Right.
so it goes back to what I said. Melanoma is what we refer to as an immunogenic tumor. The immune system actually recognizes it as foreign. Not as a wound it has to heal, but often this is foreign. And the problem with the immune system in melanoma is not, it is recognizing it as foreign. It's just not fast enough. It's like close in the race, but the cancer is ahead. And so really in that context, you just need to give it a boost. If you can give the immune response the boost it needs to kind of not exhaust and just win that race, then yeah, we've shown that with even single agent drugs, we can cure melanoma with immunotherapy. Unfortunately, I don't think that is going to work for most tumors where the immune response is either not involved at all or is part of the problem. And it's certainly part of the problem in glioblastoma. And so for those types of tumors, we've got to get the immune response off the starting line. And I think to me, that is the most impressive thing about what we showed. It really is early days for these tumors. The most impressive thing is we can get the immune response off the starting line. You can reset it. You can get it to recognize these tumors as foreign. But I do think we have a ways to go. We have a ways to really now turbocharge that response quickly and synergistically and adaptively against that malignant evolution. And in order for us to be there, we need new tools. We need to be able to combine those new tools, and we have to do so in a way that can be used where the burden of disease is as minimal as possible. So I do think you almost have to use these approaches in the context of minimal residual disease settings because of how aggressive these tumors are and, quite frankly, how behind the immune system is. And so the idea that we can generate these responses, to me, is the first step. And if we can now take the first steps, yeah, maybe we can combine it with checkpoints. Maybe we can be able to use other tools to really start to see some of the same effects we're seeing with melanomas, with single agent therapies.
We're kind of like where penicillin was when it was first
Yeah,
invented, right?
yeah, it's a
But
great analogy.
but things are moving very rapidly now, so hopefully in the next few years, we'll see some really good progress. How?
Penicillin is a great analogy because penicillin works really well against some bacteria that are just so weak or not evolved. And we're even seeing today some bacterial species that are really resistant. You need many, many antibiotics
Right.
together, combined. Cancer is a lot like that. And I do think the immune response has to kind of be that robust for some of these more aggressive tumors.
How are the parents taking it for these clinical trials? Were they eager to enroll their child in this, or was there a lot of hesitation?
Oh my gosh. Pediatrics and adults are like night and day for me, at least with this trial. I mean, I get emailed. I mean, I feel like fairly routinely from family from parents who really want this to move forward faster than it has. I mean, it's been a very slow go. And I find the adult patients are actually the ones that are more anxious and uncertain about, and appropriately so, because this is brand new. It's never been done before. There's a lot of uncertainty. And in fairness, that these patients have to be monitored in higher acuity settings. It's certainly an inconvenience to family members, patients, most importantly, who are getting this. And so that is a challenge for a lot of elderly patients who have a really a terminal disease.
Right.
Um, where in the pediatric community, I feel like it's like this can't be available fast enough. And yet I almost want to slow down that. I mean, I just want to caution that community. Like there's a lot
Right.
we don't know. We have to be careful. And so, I think, look, families are desperate, and I think people really want hope. And I have no idea if this is going to help people. I really don't. I have no idea. For all I know, it's going to just hurt people. But it does provide hope. This is a new way of approaching the war on cancer. It's a new way of activating the immune response. There is a lot of promise across our work in small animals, large animals, and even in the small cohort of adult patients we've treated. And I think as a pediatric oncologist, I'm just inspired by what that hope can mean for people, what that hope can mean for communities, even for people who have suffered the most unimaginable, most horrible thing, which is the loss of their child, what that hope can mean for them, what it can mean for their fight, to me is so inspirational. And I always say, if we can fight and we can hope and we can believe as much as these families and these patients do, we will win this war. And I have found that to be, one of the most amazing blessings of this work, is to have the partnership with this community.
I give you lots of credit because emotionally, this must be incredibly draining for you. I remember when I did my pediatrics rotation in medical school, one of the reasons that I couldn't go into pediatrics was I got very upset when young children were getting sick and having diseases that we couldn't treat. How do you deal with that on a daily basis?
I don't think I deal with it that well. I mean, I think, pediatric oncologists have to be really walk this fine line. You have to be able to be present for your patients. You have to be able to hear their stories, and you have to be able to hold their hands and walk them through this most unimaginable thing. I mean, I have 3 kids. It's the most unimaginable thing.
Yeah.
At the same time, you have to be able to not be so emotionally connected to this that you've lost your objectivity. You have to be able to, and you also, it's self-preservation. You can't be falling apart so that you can't do it again. And you have to be able to do it again and again and again. And I'll be honest, I've never been able to strike that balance really well. If I could have, I don't know that I would have ever gone back to graduate school. I don't know that I would have ever pursued a completely new approach for targeting cancer using the immune system. I mean, I think only because of my inability to kind of balance this as well as I probably need to, that I've been able to kind of trailblaze in the research field, which has its own issues. I mean, I'm now terrified about whether this is, I so desperately want this to matter to people.
Right.
Um, because of what you described. I mean, I can go on and on and on about my experiences with children, with families, with hopes and dreams that I feel responsible for failing them when we couldn't save them. Uh, and I mean, I also think I've seen things over my time as a pediatric oncologist. I mean, CAR T cells weren't around when I had children dying of leukemia. And to know what that could have meant to some of these children and just it not having arrived fast enough. And that's hard. That's hard to think about in hindsight. But then all those children we've saved now with CAR T cells, all those patients that are receiving immunotherapy that have outcomes that they wouldn't have if not for these innovations is motivating to continue to trailblaze, continue to create new tools for diseases that really haven't had anything. So it's hard. Believe me, it is really, really hard. But I'll tell you, research has saved my career, at least the ability to feel like I'm trying to do something about this. And I'm not saying everybody needs to do that, but for me, it's at least satiated that feeling of inadequacy that I have felt palpably as a pediatric oncologist.
Oh, that's great. Now, if people are interested in donating funds to help fund this research, can they do that, or is this just publicly funded by the university and the government?
Well, I mean, funding is critical for I mean, we have federal funding from the government, but that's not enough. I mean, federal funding to me is just more of a seal of authenticity that you've been reviewed by your peers in the most preeminent manners, and the work has been deemed meritorious. Um, statewide funding is important, institutional funding is important, foundation funding is important, and we've really needed all of that to move forward first in human trials. I mean, it's the cost of these trials when you're translating it from bench to bedside really is enormous. And philanthropy. I mean, philanthropy has been really the lifeblood of much of what we do, and a lot of the reason for that is there's you have discretion over that in many ways. When you have a grant, it's very clear, you have very clear aims and deliverables, and every projects can't overlap with other projects, so it ends up being a ton of projects, and I sometimes really struggle with how am I going to move forward all of these projects. So when you have philanthropy, it's more of just kind of having funds to help you get everything you need to get done, and you almost can't in the absence of it.
So if people watching this program are interested in giving you funds or money, how do they do that? Is there a website or
Yeah,
an
so the
email
University of
address?
Florida has a foundation. It's the UF Foundation, and the UF Foundation, monies that donated to the foundation, and I mean, you could even write in who or what research, and that they ensure that those monies are utilized for those purposes.
Great. So I'll
Um,
post that on this program so people watching it, if they want to donate funds, can do that.
Absolutely, yeah, the University of Florida Foundation.
That's great. Well, this has been very educational for me. I learned a lot, and I really want to thank you for taking the time to come and speak with us today. And I feel like you're about to make an incredible discovery, which will be immensely appreciated by people all over the world. So
Thank you.
keep up the good work.
Thank you. I hope so. Thank you for your time. Thank you for the opportunity and the platform. And this really has been a tremendous partnership of so many people. And I'm honestly just blessed to have these partnerships and be able to share our journey. So thank you for the opportunity.
You're welcome. Thank you.